Okay, maybe let's go ahead with the intro, and then probably we'll get some more folks coming in. So welcome to another webinar hosted by the 4D BGC working group. So we are a working group sponsored by SCOR, the Scientific Committee on Oceanic Research, and our main goal is to coordinate the development of four-dimensional data products from biogeochemical Argo observations. So we have a webinar series here to basically with an objective of sharing information on new data products that are being developed and ways that people are using them in their science. So we have a great speaker today, Louise Delaigue, who is going to be talking about the biological carbon pump. Let's see. So just some quick some quick information on the group. We formed a couple years ago. It's a photo of the group at EGU in April this year. And I mentioned our role is to facilitate discussion and coordination among groups developing, validating, and distributing 4D BGC data products with an ultimate goal of enhancing access and utility of BGC Argo data. One main thing I wanted to highlight from the group is that we have a data product archive online now, and this is available through the Biogeochemical Argo website, and it houses a bunch of different 4D data products and displays information about them and access links, and I encourage you to check that out. We also have a group website where you can find basically all of the group's activities and contributions listed. So I encourage you to check that out as well. You can join our mailing list. There's a link from the website to receive updates on future webinars like this one and other group activities. And past webinars are posted on our YouTube channel with the link shown here. Pretty easy to find if you just Google 4D BGC YouTube. Okay, with that, I'll turn it over to our speaker today. Louise is going to be talking about shifts in the ocean's biological carbon pump discovered using 4D BGC data products. So I will stop sharing and turn it over to Louise.
Thanks, John. I will start sharing. Let's see if it works. Where are you? So just give me 1 second.
That is looking good from our end.
So, oh no, I spoiled you. There you go. I just want to see my screen at the same time. Okay, cool, thanks. Let me know if the bandwidth starts being wiggly and I'll turn off my camera.
Will do.
So, thanks for giving me this opportunity to speak. So my name is Louise Delaigue. I'm a postdoc at the LOV in the south of France. And today I'd like to speak about the secret shifts in the ocean biological carbon pump. So to understand why this matters, let's take a look at how the ocean fits in the global carbon budget. So here you can see the evolution of global carbon fluxes since the Industrial Revolution. So at the top in gray and orange are the emissions from fossil fuel use, land use change, which are both increasing sharply over time, which should be no surprise to anyone. And then below zero, you see how these emissions are partitioned among the main carbon reservoirs. So you have the blue area, which represents the CO2 accumulating in the atmosphere. The green shows the land sink and the teal shows the ocean sink. So the ocean has been taking up roughly a quarter of CO2 emitted by human activities each year. That's a huge climate service, but it's not a simple passive process. So this uptake relies on a set of interconnected mechanisms that we call pumps. And one of the most important one is called the biological carbon pump. So the biological carbon pump, and I'll abbreviate to BCP so I don't butcher those words for the rest of our presentation. The BCP represents the biologically driven component of the ocean's carbon cycle. So at the surface, you have phytoplankton taking up CO2 for photosynthesis, converting it into organic matter. And part of this organic carbon is respired near the surface, but a fraction of it sinks to the ocean interior, where it can be remineralized, mainly by microbial activity, producing dissolved inorganic carbon, or DIC. And this DIC can actually remain isolated from the atmosphere for decades to centuries, which basically effectively constitutes a long-term carbon storage. And so on a global scale, the biological pump maintains atmospheric CO2 about 200 ppm lower than it would if it wasn't there. So given its magnitude, even subtle changes in the pump's functioning could actually strongly influence the ocean's capacity to store carbon. But the thing is, despite its importance, the BCP remains one of the least constrained components of the global carbon cycle. So here, I'm showing results from a paper from 2022, which summarizes projections of export production from CMIP6 model assemble. And what's clear is that there's a very large spread among models, so both in the historical magnitude and in future trends. And some show a substantial decline in export flux by the end of the century, and others actually predict little to no change. And so this wide range primarily reflects uncertainties in how models represent biological processes, remineralization depth, and nutrient climate feedbacks. And so part of the reason also for this large model spread is that the BCP remains very poorly observed, especially at the global scale. For example, direct measurements of export and remineralization are extremely sparse, particularly in key regions like the Southern Ocean, or even subtropical gyres. And this is a real problem because if the efficiency of the ocean's BCP were to change even slightly, it would directly affect how much carbon the ocean can store. And if the ocean's uptake efficiency dropped by just a few percent, atmospheric CO2 would rise much faster. So then the big question becomes, how stable is the ocean's biological carbon pump? And if it's changing, where and how can we detect it? So as I said, a major part of the uncertainty in the BCP's fate comes from a lack of observation. And to address some of the uncertainties, we can now take advantage of new global biogeochemical machine learning products. And one of these is this new GoBio2 product from Sharp et al. 2023, which provides global oxygen fields at monthly resolution over the past 2 decades. And so it's based on a bunch of different SHIP and BGC Argo data. It's merged with other oxygen data sets and reconstructed using data-driven mapping techniques that maintain temporal continuity and large-scale coherence. And what makes GoBio2 particularly valuable is that its global coverage and temporal consistency. It gives us this continuous 4D view of ocean variability from the surface to about 2,000 meters across the whole ocean. So instead of relying on sparse bottle data or model outputs, we can now use GoBio2 as a data foundation for reconstructing other biogeochemical variables, including the carbonate system. And this is really handy because once we have a nicely gridded data set that includes temperature, salinity, and oxygen, we can actually use empirical algorithms like canyon being content, which are exemplified here, to reconstruct the rest of the carbonate system on the same grid. So these algorithms essentially learn the multivariate relationships between core physical variables and carbonate chemistry, and then it allows us to kind of estimate quantities like DIC, alkalinity, PCO2, just from temperature, salinity, and oxygen. And what's particularly useful is that they also propagate the uncertainty from the oxygen field that we feed it into the carbonate system calculations. So when we apply them to a global continuous field like GoBio2, we can actually reconstruct the full CO2 system globally, monthly, and over the past 2 decades. And so in other words, we're taking advantage of the 4D structure of GoBio2 and we're extending it into common space, effectively creating an observationally constrained, time-evolving reconstruction of the ocean's carbonate system. So this is a quick look at what the DIC fields derived from GoBio2 using canyon bean content, what it actually looks like. These maps show the reconstructed DIC distribution at 4 depth, from 10 meters at the surface down to 1,000 meters. And we see the expected large-scale gradients, so lower DIC in warm, well-ventilated surface waters, a higher DIC at depth in regions influenced by remineralization and older water masses. So the patterns are basically physically consistent with what we know from independent data, from independent models, which gives us basically confidence that this oxygen-based approach is capturing what we want. It's not nonsensical. So it's pretty good for what we want to do next. So now that we have our nicely gridded DIC field, we can actually start to decompose it into different components, because what we get from the algorithms from canyon bean content is the total DIC, but that total DIC is actually made up of several distinct contributions. So we can separate it into the soft tissue component, which basically reflects our BCP, the carbonate component, which represents the carbonate counterpump, and the anthropogenic component, which is basically associated with the oceanic uptake of human-emitted CO2. There's also a smaller disequilibrium term, but our focus here is on the BCP, so we'll leave it to the side for now. So to estimate the soft tissue component of DIC, we relied on red field stoichiometry. So it's a well-established relationship between changes in oxygen, which we have, and the remineralization of organic matter. And we're using the formulation from Anderson and Sarmiento from 1994 with this canonical ratio of phosphate, nitrate, carbon, and oxygen. And so in our case, because we have oxygen from GoBio2, we can use this stoichiometric relationship to infer the corresponding changes in our DIC soft, which is our BCP. So it's basically deriving the common signal associated with the BCP. And this approach allows us to track biologically driven common variability, even in regions where direct carbon observations are sparse or unavailable. So now that we've separated the total DIC into its individual components, we can actually look at the global mean change in each of them over our study period, which I never wrote down, but it's the last 2 decades from 2004 to about 2024. So this figure here shows basically the vertical trends in DIC. So in orange is the total signal, so it's what we derived from our algorithms. In black, it's the anthropogenic component. In dark green, we have the BCP component, and then in light green, we have the carbonate component. And so as expected, we see a strong increase in total DIC throughout the upper ocean. That's not very much a surprise. That's what we expect. It's mainly driven by anthropogenic CO2 uptake, which we also expect, and which obviously dominates the signal and explains most of the vertical structure. What's interesting, though, is that the BCP component, which is our DIC soft here on this graph, remains close to 0 on average across the entire water column. So there's no clear global trend, which suggests that the overall magnitude of the BCP has remained roughly constant over the past 2 decades, again, as a global average. And so if that was really the case, if the BCP hadn't changed at all, then spatially we would expect something like this, and an empty mat, no regions of increase, no region of decrease, meaning no significant redistribution of biologically stored carbon anywhere in the ocean, which I'm sure, spoiler alert, you know is not the final result. But when we actually look at the special patterns of DIC soft, of DIC BCP, a much more complex picture emerges, because in this case, we have red areas, which indicate regions where biologically stored carbon has increased over time, while blue areas show decreases, that is a local weakening of the BCP. And what stands out is the strong regional contrast. So you have decreases across the equatorial Atlantic, Indian Ocean, even a bit in the subtropical Pacific, which is consistent with increased stratification and reduced nutrient supply. And then you see increases in the Southern Ocean and some high latitude regions, which likely reflect enhanced mixing and biological export. So even though the global mean shows no clear change, we can see that there's actually a lot happening regionally. And the BCP seems to be redistributing where carbon is stored, and these regional imbalances appear to offset each other globally, leading to that near zero global signal we saw earlier. So to make sure that the trends we're seeing in the reconstruction actually make sense, that we're not cheating or forgetting anything, we compare them against independent long-term in situ observations from well-known established time series. So we won't go into detail, but I'm basically showing vertical profiles of this the deconvolution of total DIC into its components at several stations. So you have Aloha, BATS, K2, a lot of these are very well known. And for each site, basically the left panel shows the in-situ trend derived directly from total data, so in-situ data, and the right panel shows the trend from our global reconstruction at the same location. And so the comparison shows that the reconstructed DIC that we have from GoBio2 in our algorithms actually reproduce both the vertical structure and magnitude of observed trends quite well, especially for the anthropogenic component. There are some local differences, mostly in regions with strong mesoscale variability or sparse coverage, but the overall agreement is quite robust. But we still wanted to make sure we could understand where those differences came from. So we looked at how the length of the time series might influence the comparison. And so when we summarize the comparison across sites, as shown here, we start to see a pattern. So points near the one-on-one line indicate good agreement between in situ and reconstructed DIC. DIC soft in this case. And what's interesting is that the degree of agreement strongly depends on the record length. So sites with longer records like Aloha and BATS line up nicely with the reconstruction, but for shorter records, say 5 to 10 years, the trends become more scattered. So that's not necessarily a bias in the reconstruction. We think it's really about signal emergence. The long-term DIC trend can easily be masked by interannual variability. So you need about a decade or more of observations before the underlying signal starts to stand out. So in other words, this tells us that the differences likely rely on a time of emergence because it probably takes on the order of 10 to 20 years of consistent data to reliably detect biogeochemical trends in the upper ocean. However, it's not only where the biological carbon stores climate change, it's how that carbon is sequestered vertically in the ocean. So the maps here show basically two complementary diagnostics. So you have panel A on the left, which represents the total amount of biologically sequestered DIC, that is the integrated soft tissue carbon below the winter mix layer, so the max mix layer. And then panel B on the right shows the change in the depth of sequestration, which we define as the depth where 50% of that total biologically stored carbon is found. And what we see is that in many regions, the amount of carbon sequestered and the depth of sequestration do not vary in the same way. So for example, in the Southern Ocean, we see an stored DIC soft, so again, the BCP, together with a deepening of the sequestration horizon. While in other regions like the North Atlantic, the opposite occurs. And so these decoupled changes suggest that even though the overall BCP hasn't strengthened or weakened globally, the structure and efficiency of carbon sequestration are being reorganized, sort of both horizontally and vertically.
Hey, Louise.
And so, yeah.
You've had just a couple lags, so it could be helpful to turn your video off. That's possible.
Okay. Yeah, no problem. Just tell me if it's bad. All good now?
Yes, definitely.
Cool. So from the previous figures, we see 4 distinct scenarios emerging, which represent here using, for example, regions. So each panel shows how the soft tissue DIC trend relates to changes in the depth of sequestration. So that is where and how the biological pump is storing carbon. So you also see in green the global change in the BCP, which we saw at the very beginning, which is the average BCP change or the no global change that we talked about. So if we take panel A in the Southern Ocean, we observe an increase in stored DIC soft together with a deepening of the sequestration depth, a case where both export and long-term storage are being enhanced. Then if you take panel B in the North Atlantic, you have both storage and sequestration depth decreasing. So consistent with a weakening of the pump and a shallow remineralization horizon. Then in the North Pacific, which is panel C at the bottom left, we see a reduced storage, but a deepening of the sequestration depth, which suggests more efficient transfer to depth, despite smaller export fluxes. Sorry, that was panel D. I'm getting mixed up. And then in the North Pacific, we see more carbon being stored overall, but shallower sequestration, implying that remineralization is happening higher in the water column. And so basically, it's not necessarily useful to know all of these in detail, but these 4 examples, they capture the range of behaviors we observe globally, showing that the BCP is undergoing a spatial and vertical reorganization rather than a uniform strengthening or decline. So, of course, these also depend on the intensity of the change in BSE storage or in that 50% depth, but these vertical changes may actually be kind of some early warning signals. And because if the sequestration depth in some regions were to shoal above the maximum winter mixed layer depth, then the respired carbon would be quickly returned to the atmosphere instead of being stored at depth, which would basically kind of short circuit the BCP. So these emergent shifts in sequestration depth are something we really need to keep an eye on. So what have we learned before I move on to what we have, what we reflect on in our next steps? So basically, the global strength of the BCP has remained broadly stable over the past 2 decades, despite strong regional and vertical variability. Because the global mean DIC soft is close to 0, which suggests that globally, overall, there's no net increase or decrease of the pump itself. But then beneath that apparent stability lies a major reorganization with strong regional compensations and contrasting vertical responses. You have increases and decreases in biologically stored carbon, which offset each other globally, but their spatial and depth patterns are shifting. And then emerging changes in sequestration depth could signal a shift in long-term carbon storage efficiency, because if the remineralization horizon shoals above the winter mixed layer, then respired carbon could be rapidly returned to the atmosphere, which could also be some sort of potentially early warning for the future resilience of the pump. So far, we've focused on what we can robustly infer from the oxygen-based reconstruction, that the global biological carbon pump appears stable on average, but reorganized regionally and vertically. But of course, those past 2 decades, those 18 to 20 years, is relatively short window compared to the turnover times of deep ocean carbon. So some of the patterns that we see might still reflect internal climate variability, things like ENSO or NEO-driven changes in circulation. So, of course, we're thinking of expanding this analysis to include maybe other oxidant products or a longer time frame, which might help us distinguish those natural fluctuations from externally forced trends. Also, mechanistically, you're going to be disappointed, but we can't yet say for sure why these regional differences occur. It's probably a combination of circulation, export and remineralization changes, and the relative weight of each process likely varies by region. Another point worth considering is that our reconstruction is based on oxygen, which is both a tracer and an active player in the ocean's biogeochemical cycle. So some of what we interpret as changes in the BCP could also reflect shifts in the oxygen cycle itself. So for example, changes in ventilation, respiration rates, or even oxygen utilization efficiencies. But I mean, ultimately, those oxygen-driven changes are part of the same coupled system. So if oxygen distributions are reorganizing, that still implies a reorganization of the BCP and remineralization processes that sustain it. Basically, it's the chicken or the egg story. And so to better disentangle this effect, we'll need to connect our analysis probably with some sort of complementary data sets, like things like POC export estimates or nutrients, or maybe some stratification diagnostics to try and test these mechanisms directly in models. And then finally, the fact that we already see vertical shifts in where remineralization carbon accumulates may be an early warning, as I've said. So if sequestration depth continue to show in some regions, that could eventually reduce long-term carbon storage efficiency. So the next challenge is to bring these observational constraints maybe into like Earth system models to see whether similar reorganizations appear, whether they foreshadow larger scale changes in ocean carbon storage. So there's still quite a bit to do. So yeah, that's where we are for now, globally stable, regionally and vertically shifting BCP. So this work is currently available as a preprint if you'd like to take a closer look at the details or figures. It's really a first attempt to use globally gridded oxygen products to infer those large-scale changes. I think it shows how powerful these observational data sets can be when you combine them with already existing algorithms. So of course, there's still a lot to do, linking these patterns to mechanisms, extending the record and so on, but hopefully this kind of gives us a framework for monitoring how the biological carbon pump evolves in a changing ocean. So thank you very much for listening. I'd be happy to take questions or discuss anything.
Thanks, Louise. Yeah, we'll definitely take questions if anyone has them. Feel free to raise your hand or put something in the chat if that's easier.
People have ideas of why we see what we see. I'm also very eager to take that.
Yeah, suggestions too. Let's go to Yui.
Hey, this is Yui from Mabari. Great talk. Thank you. Maybe I missed it, but can you explain a little bit more how you got the DIC soft component? I know you said there's Redfieldian ratio, so are you looking at the AOU changes over time and assuming any change in AOU is due to remineralization? Is that what you're doing?
Yeah, that's basically we used a simple approach like that. Yeah, you just multiply your DIC by the ratio between carbon and oxygen. And we used AOU. We calculate AOU from the oxygen from GoBio2, and then we derive the DIC soft from that.
Okay, got it. And then so then one part that I'm still trying to wrap my head around is that the delta DIC, the soft, is positive all the way to the surface, right? So by definition, if export, if DIC is increasing at depth, that means that there is more remineralized, right? So that means there's more export, which means that there had to be, like, the surface and the depth have to balance out to zero.
Yeah.
Because the carbon, but the fact that the line is all positive, that means that because you're using AOU at the surface, that breaks down. So that number at the surface is not that reliable above the mixed layer. Can I interpret it that way because oxygen re-equilibrate?
Yeah. But we only used DIC below the mixed layer though. So we used the winter maximum as our threshold for the MLD basically.
Got it. Okay, so the surface values so you're just focusing on depth, and then so this can't infer productivity changes, just the remineralization side of things.
Yeah. I mean, it's a good point, and I knew that was going to be sort of like one of the first questions. Like using AOU is one methodological choice. I've participated in another study where we also had nitrate data, for example, because you could have fun and try and calculate DIC soft using any of these from the Redfield ratio. And you have some differences using nitrate and oxygen. So you have kind of the same trend, but the intensity of the trend depends on which variable that you use. And I was going somewhere with this. But yeah, so using AOU is definitely a choice. I don't know if that's answer your question.
No, I was just clarifying the methods. That. Yes.
Yeah. Of course, yeah, no problem. But yeah, I know where I was going. I remember. But so the problem is that when you use those tracers, so depending on which cycle that you use, one cool/uncool thing is that because you don't necessarily get the same answer depending on the variable that you get, you kind of you can kind of infer that some biogeochemical cycles are actually changing, which is kind of what I was hinting at. Like, if you don't get the same response from using oxygen when you're trying to deconvolve your DIC, then it might also mean that these don't necessarily match anymore in a way when you look at the long-term trend. So that's also a difficulty and something else to check for sure.
Makes sense. I have a follow-up question, I guess. Slightly unrelated. Have you tried using other algorithms like Esper, to do this and see, like how much of that signal is due to Canyon B mapping over time versus other algorithms?
No, I haven't tried yet. I have a few ideas of what to do with Esper. I've spoken with Brendan Carter, who's kind of in the development of that. So I haven't actually used Esper, but it's been said to me that actually Canyon and content, with the way that it manages this decimal year business, might be a bit more accurate than Esper anyway. So we could still do a check with Esper, but apparently from what we're trying to constrain with the existing algorithms, this is as good as it gets. But I'd be keen to look at the preformed DIC from Esper, for example, because I don't think I can do that with Canyon. So I think it'd be cool to look at that as well and compare that to the trends that we see with the biological DIC.
Frank Muller-Carger has put in the chat a question. So I'll let Frank speak up if he wants to, or I'll just go ahead and read it.
I'm
Yeah,
reading
hi,
it. But
Louise. Hi, John. Thanks for
okay,
Frank.
And I'm really sorry I arrived late. Typical, another meeting spills over. But I arrived at the time when you were explaining some of the regional changes and the vertical changes over time and on a rate of meters per year.
Yeah.
And I wonder with the issue that we have with gappy sampling and time and space, how good those estimates may be, or it's a number, but just any small seasonal sampling bias may throw those numbers off. So I just wondered how you characterize the uncertainty or the significance of those rates.
Yeah, thanks. No, that's an excellent point. So I haven't done an uncertainty analysis for those depths yet. I'm trying to figure out how to do that. The thing that's a bit tough is that's why I was trying to explain the intensity of the change is not the same everywhere. Like it's not like I'm finding like a global trend of X meter per year difference everywhere. So to try and do an uncertainty analysis on the full data set, I haven't very much found out, because usually I do like some, because I've done a lot of Monte Carlo uncertainty propagation for this analysis, and for the depth, yeah, you don't really have like you don't really have like a depth uncertainty from like CTDs or like stuff like that. So this is still something I'm very keen to have suggestions if you have any. I also think that's something that we need to deepen as well in the analysis for sure.
Yeah, I mean, if you can, maybe as the simplest first cut could be seasonal, look at seasonal trends and in both hemispheres when you have deeper mixing and stratification, and then look at a compiled map where you combine those and some seasonal composites.
Yeah, because I guess we simplified our lives using the winter max layer, so winter maximum mix layer.
Yeah.
So, but yeah, no, that's a good suggestion. Thank you. I can definitely look into that.
I was curious while we're waiting to see if anyone else raises their hand. So you showed the export flux from different models, I think, as a time series.
Yeah.
And there's like huge variability, not just on the trends on those, but on the magnitudes. The one you showed of the like the model time series of export flux, I think.
This one?
No, toward the beginning with like the different cement models.
What did I This one, yeah.
Yeah. Yeah.
Yep.
So I mean, I was struck by, not only the changes that there's quite a lot of variability in, but the magnitudes themselves, and there's huge spread in the magnitudes of export flux. So can you comment on like how these sorts of observational constraints might help bring that spread kind of closer together?
Are you asking me to solve, the biggest problem in estimating the end of the century BCP export? Yeah, sure, John, I'll just do that right now. No, well, first of all, my first impression looking at this graph was, wow, we just basically, do we know anything, because it's very impressive, this range. I agree with you. I think because there's actually a working group that's working on this that's gonna meet after Ocean Science in February. I don't know if you're part of that at all, but I'm part of a couple of working groups that are attached to that workshop, and sometimes it's as simple as including TEP, like the TEP from planktons, like that sticky goo, which I'm not entirely familiar with, but that also aggregates carbon throughout the water column, and that's gonna, I think, matter a lot for that export flux. So I think it's very difficult in the sense that it's not just like of course in this analysis, like it's having this kind of simplified way of looking at dissolved DIC and trying to infer the biology, but the biology itself is also governed by so many different very tiny processes that I think we're just having a hard time reconciling everything. But I'm sure that's a well-known fact. This isn't something new to anyone here. So yeah. Also, thanks, John, because it is Go Bio 2 after all. So I feel like I should give you a shout out because there would be no analysis without that product. So
Yeah,
so it was thanks to John as well,
Happy to see people using it. Okay, any other questions for Louise?
I mean, people can also get in touch afterwards if they get super excited about it. Sleeping at night. My email address is at the bottom of that slide if anything comes up.
Awesome. All right, then I think we will wrap things up there.
Cool.
I want to mention we also have a upcoming webinar in December. So look out for updates about that. If you're not on the mailing list, you can go to our website and join. And yeah, I think that's it for today. So thank Louise again, and thanks everybody for coming.
Thank you.
Bye all.
Bye.
Thank you, Louise.
Bye. See you.